too long, and it's a good introduction. We do have to start talking about it. Okay. So our unit is on biology, all right? And the things that we cover in this biology unit are tissues, organs, and systems of living things. So to start with, for the first couple of weeks, we're going to talk about cells themselves, those little tiny units of life that we're going to define here in a moment. But in the end, when we're finished this, and we'll likely finish off just after Christmas, in the end, we're going to be talking about the systems of a living organisms. For example, like the respiratory system, right, that we use obviously as living things to bring air and oxygen inside our bodies so that our cells can use that stuff. So we will ultimately be talking about the most complicated kind of systems that our bodies have. The musculoskeletal system, our muscles in our skeleton that allow us to move. Our nervous system, the nerves and our brain and how it functions. So we're starting off with the simplest parts of a biological system, the cells, and we will be working our way up to the more complex systems that we see in living organisms. Okay, so what makes something alive? So I do want you to write that down. What makes something alive? So we're going to start by talking about living things. And I could ask you what you've learned in the past, but I'm sensing a kind of a Monday morning blah, and I'm not necessarily thinking you guys are going to be offering up too many ideas. So I'm going to just put them out there. What makes something alive? Living things have a lifespan, all right? So living things will have a birth or a beginning. There will be some kind of time within there where that living thing is doing what it's kind of I see again designed to do. I don't know. Does anybody design living things? What its kind of function is, and then ultimately that living thing will cease to live, and there will be death, and there will have a complete lifespan. So living things have a lifespan. All right. Am I good to go? I have no idea what we got here. Number 2, living things grow in size, they reproduce, and they are able to repair themselves. All right? And this kind of idea here that there's growth, reproduction, and then ultimately repair is why we have cells. Right? I'm kind of jumping the gun, but I think you guys all know that we're made up of tiny little units called cells, skin cells and muscle cells and all that type of stuff. Cells allow us to do all these things. The reason a baby grows to being an adolescent and then ultimately an adult is because more and more cells are added to the system. You got more cells in your bone that make the bones grow, more cells in your muscles that make the muscles grow, etc., etc. Reproduction, right? You guys all, again, out of my league a little bit, but you all take health and phys ed. You've all had your discussion on reproduction. Cells involved in those things. You need a sperm cell from the male. You need an egg cell from the female. Because they're cells, they can come together to complete that cell and then ultimately produce another living thing. So again, cells allow this all to happen. Repair. You end up with a gash in your arm, some blood cells leak out. Well, if you were made up of only one cell and you couldn't repair that, you had only one big cell, all of your interior would leak out, you'd be dead. But because we're made up of many cells, these other cells come in, these platelets, they form a clot, they stop the bleeding, white blood cells come in to take care of any infection, and ultimately new skin cells are formed, and you're back in business and you're back doing your thing. So cells are what allow all of these things of a living thing to take place. Okay, number 3. Living things can sense and respond to changes in their environment. When we say environment, we're not talking about like environmental issues. You know, it's not like we're talking about global warming or anything like that. We're just saying what is around the living thing. So as humans, our ability to sense is quite complex, right? We've got all kinds of different senses that allow us to see what's around us and then be able to respond to those kind of stimuli that are there. Here I've got a little bunny rabbit being chased by a lynx or a cat of some sort. So whether that bunny rabbit responded fast enough or not, I don't know. I don't know how this ends. You can imagine that however you'd like. All right, so living things can sense and respond to their changing environment. Living things require energy. Now, it's not part of the curriculum to talk specifically about plants in this unit, but I will be talking a little bit about plants. Plants are living things. They can do all those things. They can respond to the environment. Like if there's more light in one area, they can kind of bend and turn towards the light. So living things require energy. Plants, they tend to use the sun's energy to make sugar so that they can use that in order to grow and have more cells. Animals like ourselves and other regular animals, they do not have the ability to use the sun's energy, so we need to consume sugars that are produced by plants or eating other animals, right? That type of thing. But that is required in order to maintain life. Living things produce waste. And yep, there's part of the stuff that we take in that is not necessarily useful to the body, and so that gets expelled as waste. We will talk more about that in gory detail when we get to the digestive system. Right now, we'll just say there's waste produced. I guess exhaling carbon dioxide, that's waste. It's not as gory as the waste you probably were thinking about, but that counts. That's a part of a molecule that we don't use, so we exhale that carbon dioxide. Okay, so let's talk a little bit about cells. Because as I was implying in those previous slides, cells work very well in maintaining all of those things, as well as explaining all of those things. We can explain why we have cells so that we can repair, so that we can reproduce, so that we can grow. Historically speaking, and I don't know quite as many stories about biology, but I think we're talking about the time of Sir Isaac Newton, so we're talking about early 1700s. It's about the same time as when the telescope is invented, and guys like Galileo are looking at the stars and looking at Jupiter and seeing the moons of Jupiter with their telescope. Similar guys, and I can't remember the name, I want to say Van Leeuwenhoek, something like that, a Dutch scientist, created a microscope, and then a guy by the name of Robert Hooke, an Englishman, used that microscope to look at all kinds of things and ultimately they realized, hey, there are these small little repeating units in all of these examples of living tissues and living things, and so we're going to call them cells, and the rest is kind of history. So we're talking about early 1700s where these ideas are starting to come about. Before that, the scientists would have no knowledge of the fact that there were these little pieces within all of us. They didn't have the ability to see them. So what did they conclude after they looked at a whole bunch of stuff? All living things are comprised of one or more cells. All right, so they're made up of these cells. The cell is the basic unit of life. So it is the simplest little thing that you can extract from something and say, that thing is alive. All right? Now, things like viruses, which we're not talking about in this course, I don't believe, they're smaller than a cell. Does a biologist consider a virus to be a form of life? It can reproduce. It does need energy. I don't know. See, there's the type of answer that I really am not sure whether you'd say virus is a form of life. It has the ability to reproduce. It does use energy, but it doesn't have the ability to reproduce necessarily on its own. It's got to use another cell. It probably is. I really don't know though. This is where my knowledge is a little more limited than in the other subjects. But that's okay. We could always look that up. But again, viruses are not necessarily part of this course. So at this point, we're saying the cell is the basic unit of life. It's that kind of most simplest piece that we can talk about as being alive. Okay. All cells come from pre-existing cells. So there has to be some kind of cell division that we're talking about here. So you've got one cell, it does its thing, it lives its life as a cell, and at some point it divides and it becomes a pair of cells. All right? This is a form of reproduction. It is different than what you would consider from your health class as reproduction, eating a sperm and an egg. This is different. We'll contrast that when we do talk about this later on. Yep. Oh, it's not like you're cutting it in half. The cell grows to the point where it then becomes in a state where it can divide. It divides, you end up with two smaller cells, and then they grow again to their proper size. So it's not that we're actually just cutting them with a knife and getting smaller and smaller cells. Well, you could cut a cell in half, but that wouldn't be the same as what we consider the production of new cells. That would be cutting a cell in half. That would be different, right? That would be destroying the cell for the sake of looking what's inside, where in this case it grows and then it splits into two completely functioning cells. Okay? Okay. Okay, let's talk about the microscope. Now at this point, hold on, don't write that down. Some of you people are writing like crazy. What do I want you to write? I do want you to write that down. I do. And then once we're done writing these down, then we will get to, oh, I want to go backwards. Back up, back up, back up, back up, back up. Okay. The compound light microscope. So at this point, we will write this down. Yeah, you will. And then I'm going to show you the microscope as we go along. And then when I'm done that, my video will be done. You will then work on a sheet that is going to label the microscope and go over its parts. All right? And I'll talk a little bit about their parts. And then we'll take the sheet up. And once we're done that part, then we will get into talking about how we're going to do our pictures and all that type of stuff. So I am going to get you to write this down. So here I've got a cart full of microscopes. All right? And I'm going to grab one here. When we move our microscopes, one arm firmly on the back like this, other hand on the bottom so we don't drop it because they're about $500 pieces of equipment, and you bring it to where you're going to be working, okay? All right, so the reason it's called a compound microscope is because it has more than one lens being used. It's not like a simple magnifying glass. All right, so let me bring this whole thing up. And you can write that down. And then I'll talk about some of these parts here. And I'll talk a little bit about magnification and then you'll be able to do that worksheet that I'm talking about. So most cells are small, obviously. There are exceptions, like an egg, like a chicken egg. That's a single cell. So they can be large, but they tend to be small. They tend to be measured in what we call micrometers. Now, I've got an issue with my font here. This U actually has a little tail on the front of it. It looks like this. It's a U with a line on the front end. You could imagine that it almost looks like an M. Now, that little tail on the front end in the metric system stands for micro. So a micrometer means that it's well, you know what? That is a completely wrong statement there. And I've been teaching this for I don't know how many years. 1 micrometer is not 1,000 millimeters. 1 micrometer is 1,000th of a millimeter. I need you to put there 1 divided by 1,000. You see what I got there? Put the 1 in front of it with a divided by in front. It's 1,000th of a millimeter. It's a tiny little fraction of a millimeter. So it would take 1,000 micrometers put in a row to make 1 millimeter. Does that make more sense? It's tiny. Micro. That's why we use that prefix. You can think of it in terms of kilometers. It takes 1,000 meters to make 1 kilometer. So 1 meter is 1,000th. It's a little piece of it. So a micrometer is 1,000th of a millimeter. Okay? It takes 1,000 of them to make 1 micrometer. So that's why we call it a microscope. When we look in at those cells, we're looking at those very small dimensions. A compound light microscope allows light to pass through a thin specimen and combines the magnification of 2 lenses. So you know how we used a magnifying glass to make something bigger? That I know how it works. A microscope uses 1 lens, like a magnifying glass, and then a second lens to magnify the magnified image. Right here, I can show you those 2 lenses. So in here, we've got this revolving nose piece. Right here, this revolving nose piece. And you can see it there on the picture. Right? On the revolving nose piece, there are 3 lenses, and we can interchange them. You can see we can rotate them out, and they've got a little stop when they get into the position. We put our specimen on this thing here called the stage, and these things are called stage clips. They hold the specimen in place. We put the specimen on a little microscope slide, and we put that slide on the stage, all right? And then we choose the lens that we want to use. We always want to use the shortest lens first. That's the weakest one. These are called objective lenses because they're closest to the object. Remember when we drew ray diagrams, we always had the object? The thing that we're looking at on our slide is the object. We're going to look in here and see an image. So because these are closest to the object, they're the objective lens. And we always start with the smallest one. It's the weakest, okay? So there's lens number 1. Lens number 2 is right here. Now sometimes I can take it out, but I can't. There's a lens right up here by my eye. It's called the eyepiece lens or ocular lens. Ocular simply means eye, pertaining to the eye. So you look in the eyepiece and you see the circle that is what we call the field of view. How many people have looked in a microscope before? Excellent. So many of you have. You're going to look in there and you're going to see a circle. We call that the field of view. That's what you can see. All right? And So what we do is we get our object on there, and we adjust it to the lowest one, and we take a look in, and we adjust the focusing using this coarse adjustment knob. Notice the coarse adjustment knob moves this whole tube up and down. Can you see how that's working? Moving the tube up and down? That brings it closer and further away from the object so that you can see it. Once you get it nice and clear with the coarse adjustment knob, you can switch it to the second one, the second most powerful one. You'll now zoom in. You won't need to use the coarse adjustment knob anymore because you're already in the right spot. So to make it focus now, you use the little wheels, and the little wheel is called the fine adjustment knob because when you turn it, it moves it just a little bit. You actually cannot see the tube move when I adjust the fine adjustment knob. You see how you can't see it? But it is moving very finely, very tiny little bit amounts. So you can then focus it under the medium power. So you always start under the low power, the short one. You use the coarse knob. You then go to the medium power, and you use the fine knob. Then you can go to the high power and use that fine knob a little bit again, and you will see a big, nice, clear image. Okay? All right? The back of it is called an arm. That's what supports it. That's how you carry it. The bottom part's called the base. These have mirrors that you can reflect the light in, but we've also purchased some light sources. Just little oh, this one doesn't work. I need a new battery. Just some low-cost light sources. Okay, do any of these work anymore? Okay, I'm really frustrated because we're going to have to use this. This is terrible. I'm going to need to find some batteries. Oh, here we got one. Woo! These are just these low-cost lights that you can like stick to a wall or something like that. So what we do here is we just place that light source on there. Sometimes it's too bright. So what I ask students sometimes to do is just take a piece of tissue paper and put the tissue paper over top of that bright light source so it kind of cuts down on the brightness. You can see it's not perfect, but it does work. It does work okay. Okay, yeah, low cost, exactly. They were like $1 each. So now the next question becomes, how do we know how much bigger this is? What's the magnification? That's the last thing I'm going to get us to write for today, and then we'll start talking about how we're going to. Oh, sorry. Then you're gonna do your thing. Yeah, you don't have to. I'm going to just tell you to do the microscope sheet. You don't have to write any of that stuff down there, but I do want you to write down magnification in the microscope. Because it's like we're using two magnifying glasses, the way you determine a magnification is really easy. You simply...
Mr. Saunders, can I have Riley Levine to the main office, please?
Yep.
Thank you.
So, to find the magnification of a microscope, it's really quite easy. All you have to do is multiply the magnification of each of the lenses. All right, so let's say I had, and I don't, not for our microscope, but let's say we had a magnifying glass that doubled the size of this, and then you put another one that doubled it again. It would be doubled, which would be times 4, right? So you simply multiply the powers of each of the lenses. Our eyepiece lens, or our ocular power, is 10 times. So this guy up top here makes things 10 times bigger. Down on this revolving nose piece, all right, there are 3 different lenses, and they will have 3 different effects. The low power is times 4. So when you put together the times 10 and the times 4, you end up with an image that is 40 times bigger than what you would be looking at with just your eyes. what would be the magnification under medium power? So let me put under here, magnification, medium. What are my two numbers that I'm going to have there? Yep. 10 from the eye, and 10 times 10. So medium power is, what's 10 times 10? 100. 100. Okay? So when you flip to the medium, you're looking at it 100 times bigger. And we might as well finish here. Why don't we do magnification of high? What is going to be the magnification of high? What am I going to put there? Yep. So it's going to be 10 from the eyepiece times 40 from the high, and that is 400 times. It will be 400 times bigger. At this point, if you're done writing, you can open up the document in Google Classroom, not the one that is the turn in for marks, but just the lesson, and I want you to label and complete the functions of the microscope, and we'll take that up in about 10 or 15 minutes. All right? Yeah, you can while you're doing that. That's fine. So again, I'm going to just show you here. I'm not talking about the assignment because we're not getting to the point where we're doing that. I'm not talking about how to draw a dry mount. I'm talking right down here, and mine is not reading the drive files very well. Oh, that's because I'm probably offline. So I don't have a preview, but notice where it says the cell theory and microscope. I think this one down here is the microscope picture. So my Google Classroom is failing me. What's that? I'll take care of all that. That's great. You just give me that whole thing, and you can do what you need to do. So at this point, you're looking just at the little document about the microscope, and you're going to label it, and you're going to then talk about the parts of them, that type of thing. So I'm going to just stop my recording as you guys are working away there.